My name is Joshua Mugabi. I welcome you to topic 8, which is growth in plants and development in insects. We are going to look at learning outcome A. Analyze the pre and post germination stages during the growth and development of plants in relation to their significance in crop production. Our learning activities. We shall look at part A. In groups, learners search for and discuss the concept of seed dormancy and seed banks, factors affecting and measurement of growth, primary and secondary growth. Let's start with seed dormancy and seed bank. Item 1. We are going mainly to emphasize on beans and maize germination. A farmer plants maize and beans at the start of the rain season. While most seeds germinate quickly, some remain in the soil and germinate weeks or even months later. Nearby, a conservation agent stores seeds in a national seed bank to preserve plant species threatened by climate change. Task. Define seed dormancy and explain why some seeds fail to germinate even under favorable conditions. 2. Identify types of seed dormancy and their causes. 3. Explain the concept of a seed bank in A. Natural ecosystem. B. Conservation and agriculture. 4. State the importance of seed dormancy and seed banks to plant survival. Let us start with task 1. Define seed dormancy and explain why some seeds fail to germinate even under favorable conditions. Seed dormancy is a condition in which a viable seed fails to germinate despite favorable environmental conditions. Identify types of seed dormancy and their causes. Types of seed dormancies include physical dormancy, where a hard seed coat prevents water or oxygen entry. Physiological dormancy. This is due to the presence of growth inhibitors such as abscisic acid. Morphological dormancy is a result of embryo being immature. Explain the concept of a seed bank in a natural ecosystem. A natural seed bank is a reserve of viable seeds present in the soil. Let's explain the concept of a seed bank in conservation and agriculture. An artificial seed bank is a controlled storage facility preserving seeds for future use. State the importance of seed dormancy and seed banks to plant survival. Prevents germination during unfavorable seasons. Ensures species survival after disasters. Maintains genetic diversity. Supports crop improvement and conservation. Still under seed dormancy and seed bank, item 2, we want to emphasize on delayed germination. After heavy rains, weeds appear in a maize field that had been left uncultivated for 2 years. Surprisingly, some weed species emerge only after the soil is plowed. Task: explain why seeds can remain in the soil for years without germinating. Identify the types of dormancy involved. Explain how plowing stimulates germination. Let's start with explain why seeds can remain in the soil for years without germinating. Seeds remain dormant due to hard seed coat or chemical inhibitors. Identify the types of dormancy involved. The dormancy is mainly physical and physiological dormancy. Explain how plowing stimulates germination. Plowing breaks seed coats, increases oxygen supply, and exposes seeds to light, triggering germination. Still under seed dormancy and seed bank, item 3. Here we are going to look at seasonal survival of desert plants. In semi-arid areas, many plants germinate only after rare rainfall events. Task: explain the role of seed dormancy in seed plant survival. State 2 environmental cues that break dormancy. Let's start with explain the role of seed dormancy in desert plants. Dormancy prevents germination during prolonged drought ensuring survival. Stage 2 environmental cues that break dormancy. Rainfall, water availability, and temperature changes break dormancy. Item 4, still under seed dormancy and seed banks. We are going mainly to emphasize on National Seed Bank. A country establishes a seed bank to preserve indigenous crop varieties. Task: define a seed bank. Explain 3 benefits of artificial seed banks. State conditions necessary for long-term seed storage. Let's start with the define a seed bank. A seed bank is a facility for storing viable seeds under controlled conditions. Explain 3 benefits of artificial seed banks. Benefits: conserves endangered plant species, maintains genetic diversity, and provides seeds after disaster. State conditions necessary for long-term seed storage. Low temperature, low humidity, and pest-free conditions. Still under seed dormancy, we have item 5. We are going to look at forest regeneration after fire. After a bushfire, grasses and shrubs rapidly regenerate. Task: explain the source of new plants after the fire. Identify the ecological importance of soil seed banks. Let's start with explain the source of new plants after the fire. Seeds from the soil seed banks germinate after fire breaks dormancy. Identify the ecological importance of soil seed banks. Soil seed banks ensure ecosystem recovery and stability. Item 6. We are now going to look at factors affecting and the measurement of growth. In this item, we are going to emphasize on growth conditions in plants. 2 bean plants are grown in different conditions. Plant A receives adequate water, nutrients, and sunlight. Plant B grows in poor soil with limited water. After 3 weeks, plant A is taller and heavier. Task: define growth in living organisms. Identify internal and external factors affecting plant growth. Describe methods used to measure growth in plants. Explain why increase in dry mass is a reliable measure of growth. Let's start with define growth in living organisms. Growth is a permanent and irreversible increase in size, mass, or number of cells. Identify internal and external factors affecting plant growth. Factors affecting growth: external, internal. Internal include genes, hormones like auxin, gibberellins, among others. External factors include light, water, nutrients, temperature, and oxygen. Describe methods used to measure growth in plants. Measurement of growth includes measuring increase in length or height, measuring increase in fresh mass, measuring increase in dry mass after removing water, or counting number of cells. Explain why increase in dry mass is a reliable measure of growth. Dry mass is reliable because it excludes temporary water fluctuations and reflects actual biomass increase. Still under factors affecting and measurement of growth, item 7. We are going now to look at unequal growth in school garden. Students grow cabbage plants. Those in shaded areas grow slowly. Task: identify the limiting factor affecting growth. Explain how light affects growth. Suggest a solution. Start with identify the limiting factor affecting growth. Light is the limiting factor. Explain how light affects growth. Light affects photosynthesis and energy supply. Suggest a solution. Transplant to areas with adequate sunlight. Still under factors affecting and measurement of growth, item 8, we want to look at nutrient deficiency experiment. Plants grown in nutrient-poor soil show stunted growth. Task: explain the cause of poor growth. Identify nutrients essential for plant growth. Explain the cause of poor growth. Lack of mineral nutrients limits cell division and enlargement. Identify nutrients essential for plant growth. Nitrogen, phosphorus, potassium, and magnesium. Still under factors affecting and measurement of growth, item 9. We are going to look at measuring growth accurately. 2 seedlings are weighed daily, but their masses fluctuate. Task: explain why fresh mass is unreliable. Describe a more accurate method of measuring growth. Task: let's start with explain why fresh mass is unreliable. Fresh mass varies due to water content. Describe a more accurate method of measuring growth. Measure dry mass after drying to constant weight. Factors affecting and measurement of growth, item 10. Let's now look at temperature and growth rate. Seedlings grown in a greenhouse grow faster than those outside. Task: explain the effect of temperature on growth. State the optimal temperature concept. Explain the effect of temperature on growth. Temperature affects enzyme activity, controlling metabolism. State the optimal temperature concept. Growth increases up to an optimum temperature, then declines. Factors affecting and measurement of growth, item 11. Let's now look at hormonal control of growth. Application of auxins causes bending of plant shoots. Task: explain how auxins affect growth. Identify the types of growth involved. Let's start with explain how auxins affect growth. Auxins stimulate cell elongation on the shaded side. Identify the type of growth involved. It's the differential growth, which is the same as phototropism. Now let's go to primary and secondary growth. Item 12. We shall start with increase in size and height. A young sunflower plant increases rapidly in height, while an old mango tree increases mainly in trunk thickness rather than height. Task: distinguish between primary growth and secondary growth. Identify the meristems responsible for each type of growth. Explain the importance of secondary growth in woody plants. Compare primary and secondary growth using a table. Let's start with distinguish between primary growth and secondary growth. Primary growth is growth in length of the roots and shoot. Secondary growth is growth in the thickness or girth. Identify the meristems responsible for each type of growth. Primary growth is caused by apical meristem. Secondary growth is due to lateral meristem of vesicular cambium and cork cambium. Explain the importance of secondary growth in woody plants. Importance of secondary growth. It provides mechanical support. It increases transport capacity of water and food. It forms protective bark. Compare primary and secondary growth using a table. So we have the features, we have primary growth and secondary growth. Direction. Primary growth is in length. Secondary growth, girth, stroke, thickness. Meristems. Primary growth is by apical meristems. Secondary growth is by lateral meristems. Where they occur. Primary growth occurs in all plants. Secondary growth occurs mainly in dicots and gymnosperms. Function. Primary growth is for elongation. Secondary growth is for strength and support. Still under primary and secondary growth, item 13. Let's now look at the young versus mature plant. Maize plant grows taller, while a tree trunk becomes thicker. Task. Identify the type of growth in each plant. Name the meristems involved. Let's start with identify the type of growth in each plant. In maize, the type of growth is primary growth. In the tree, the type of growth is secondary growth. Name the meristems involved. Apical meristem and lateral meristems respectively. That is, in maize, it is apical meristem, and in the tree, it is lateral meristem. Still under primary and secondary growth, item 14. We want now to look at ring formation in tree trunks. Annual rings are visible in a tree stump. Task. Explain how annual rings form. State their importance. Let's start by explaining how annual rings form. Rings form due to seasonal activity of vesicular cambium. State the importance. Importance. Annual rings are used to estimate tree age and past climatic conditions. Still under primary and secondary growth, item 15. We shall look at transport efficiency in woody plants. Woody plants transport large volumes of water. Task. Explain how secondary growth improves transport. Identify tissues involved. Let's start by explaining how secondary growth improves transport. Secondary growth increases the xylem and phloem tissues. Identify the tissues involved. Secondary, xylem and secondary phloem. We're still under primary and secondary growth, item 16. Here we are going to look at protective function of bark. All the stems develop thick bark. Task: identify the tissues forming bark. State its function. Identify the tissue forming bark. Cork cambium forms cork tissue. State its function. Protection against injury, water loss, and pathogens. We are still under primary and secondary growth, item 17. We are going to look at absence of secondary growth. Palm trees do not increase in girth like dicots. Task: explain why palms lack secondary growth. State the types of plants that show true secondary growth. Explain why palms lack secondary growth. Palms lack vesicular cambium. State the types of plants that show true secondary growth. Dicotyledonous plants and gymnosperms. Thank you very much for participating in topic 8, which has been growth in plants and development in insects. We have been looking at learning outcome A, analyze the pre and post germination stages during the growth and development of plants in relation to their significance in crop production. We have been looking at learning activity A. In groups, learners search for and discuss 1. The concept of seed dormancy and seed banks. 2. Factors affecting and measurement of growth. And 3. Primary and secondary growth. This has been very useful. Visit me at Mugabi Joshua Biology on YouTube.